thanks everyone for coming. So yeah, so this is an introductory session to BIGSdb. So we just describing the design ideas behind it and some ideas of the implementation and how you can use it. It's a completely informal session, so please stop me at any time if you want to, any questions. And we can also sort of do sort of more informal demos at the end if people feel that would be useful. Okay, so the BIGSdb platform, it stands for Bacterial Isolate Genome Sequence Database, kind of does what it says on the tin, really. It's designed to bring together lots of different kinds of information so that they can be analyzed together. So it really sort of derived from the earlier generations of MLST databases that we've been developing now really since the first MLST scheme back in '98. So there's over 20 years worth of development in the ideas that we're sort of presenting here. And really, the database has isolate records, which consist of metadata that describe where it comes from and some sort of maybe phenotypic information, what disease it causes, the provenance, so year, place, etc., and then various and sort of tracking information as who submitted it and when it was last modified, and then it's got sequence data, and that sequence data can be either just like the results from a single sequencing reaction, as like a Sanger sequencing run, just small single gene results. It could be the result of, say, like an Illumina sequence assembly, where you've got a draft genome that is present in multiple contigs, or it can be a complete genome, a complete single piece of DNA. As far as the system is concerned, it doesn't care. DNA is just DNA, and it's just really looking within the sequence to try and find the regions that you're interested in. So the really simple design philosophy is like this. We actually have two sides to the system. Here, so on the left-hand side, we have the nomenclature. So this is really the typing databases, and this is where we define all the loci, so the genes or regions of the genome that we're interested in, and all the identified variant sequences for each of those loci. Okay, so when we started, we would just have MLST, so there'd be 7 gene loci, and we would store every known allele that we found of those sequences, and we just give those an allele number. Obviously, that's now expanded significantly, so we've got sort of thousands of loci that we're interested in. Then on the other side of the system, which is a bit more interesting, is the isolate and the genome database. So this contains all the provenance and phenotypic information and the genome sequence, which, as I said, could be either a whole sequence or it could be draft genomes or whatever. And then, in order to identify the particular variants for each of the loci that we're interested in, then it will do a BLAST search. Now, normally when you do a BLAST query, say like on NCBI BLAST, you will have a small sequence and you'll query it against a big database of all genomes and stuff. In this case, the query is actually the other way around. The BLAST database contains the small allele sequences, and the actual query is the entire sequence bin. So you're querying the whole genome against a database of allelic variants. And if it identifies an allele sequence for a locus, then that gets recorded within the database, so that next time you don't need to scan it. It's already within the database and can be used. So conceptually, that's how it works, and that's how we originally did it. The very first version of BIGSdb, it would work. It would go locus by locus. It would perform a BLAST search for each locus, and that BLAST query would contain all the known alleles for that locus. Now, as the databases have got bigger, you can imagine that would get slower and slower. So that really wasn't a scalable solution. So now we do one big BLAST search that contains all loci together, and we also cut down the number of alleles we put into that BLAST database. We actually now only put exemplar alleles that cover all known diversity within sort of 10% identity. So it's essentially, we use BLAST now to identify the sequence region, and then we just do a direct database lookup of the of the sequence that gets pulled out, and that's much quicker than that BLAST search. So the BLAST query itself now doesn't, only has maybe 4 or 5 different alleles for each of the loci that we're interested in. And from that, we can now, the amount of time it takes to scan a genome against maybe 2,000 loci is about a minute or so. That's on one processor core. And this is running on a server that's got 120 different cores, so we can parallelize this really quickly. And so it's not this used to be a bottleneck in the whole process, but it's got a significantly quicker. And we can do about 25,000 genomes a day now on the one server. You don't normally have to do that many, but we can. So it's not really a problem anymore. That's if you were doing it on the command line as a sort of background process. Many of you may have done it through the web interface, in which case you're doing it on a single core, and depending on how you're doing it, it might be doing it locus by locus. So that tends to be a lot slower. But for the routine scanning of new genomes that goes into the database, then we'll do it on a multi-threaded and a big query searching everything at the same time. So really just to reiterate what data we are storing within the databases, the 2 main database structures. The first one is it's a typing, your nomenclature. That's everything that's on the left-hand side of that previous image. So you've got your sequence definitions, and this is literally every known allelic variant of each of those loci. Okay, so these may just vary by 1 or 2 nucleotides within the alleles, but we store the whole sequence within there. And then the other thing we store within there are allelic profiles for schemes such as MLST, whereby you will define a sequence type for every known combination of alleles for a particular set of loci. And that's now expanded out to core genome MLST, but the principle is exactly the same, but you might have 1,500 loci rather than 7. But you still define a sequence type for that particular combination. So that's nomenclature, and that's pretty boring actually, but very useful. Your other side of it is your isolate database and your genomes. So as I said already, this is provenance, so this is where it was found, time it was found, who found it, maybe some phenotypic information, some result of sort of biochemical tests maybe, you might have some serogroup information, things that you could determine without actually looking at the sequence. And or disease, disease that particular isolate has caused. And then we have the genome sequence as well, okay? So if we're just using the typing database, the system on the left-hand side of that chart, then we can use BIGSdb just as a nomenclature server. So you can query it using your own sequence without having to put anything into the database. So if you have a genome sequence or even just a Sanger sequencing run, you might have just 700 base pairs of sequence and you want to identify an allele, then you can just paste that into a web form, select the scheme, or the locus that you're interested in querying against, and you can paste a whole genome into this box, or you can upload it directly using a FASTA file, tell it the scheme you want, and it will identify those alleles. And if the known combination of alleles, in the case of MLST, is known, then it will also tell you the sequence type and maybe any other information associated with that sequence type. And that process is very quick. That takes about a second for a genome sequence. Likewise, if you've got a core genome scheme, you can do the same. It takes a bit longer. It takes up to, I would say, about a minute to do that from a pasted-in genome. And in just the same way, it will identify, because normally you won't exactly identify a core genome sequence type from a genome, because you only need one allele difference to be a new sequence type. Almost every genome has a different sequence type. So in this case, it will tell you it will find the closest match and report that, tell you how many mismatches there are to that closest match, along with percentage ID. And then we can use clustering based on those core genome STs that have been defined. And I'll go into a bit more detail later on as to how we do that. And from that, we can identify similar core genome STs based at different thresholds of differences. And we set up lots of different thresholds depending on the organism. For some of them, there's a particular reason why we choose those thresholds. We actually find there's a discontinuity in the sort of way the variation is shown within the population. For others, it's just arbitrary. It's 200, 150, 25 base loci differences. And so at those different thresholds, we can identify different profiles, so that's different core genome STs that are similar at that level of at that threshold. And then we can map that back to isolates that are in the database in order to retrieve isolates that are similar at those different levels. So if we're looking at a 200 locus difference from the genome we just pasted in, you see that there's 4,800 or so core genome sequence types, and those correspond to 3,400 isolates. And those are hyperlinked, so you can just click on those, and that takes you into the database and returns those results. As you get more stringent in your thresholds, then obviously you get much fewer isolates and profiles back. And so even at 100 locus differences, we're actually only finding one isolate that's similar at that level. And in fact, this is actually, when I did this, it was actually the same sequence that was actually returning, but it was a different assembly. So there was slight differences in the assembly, but it was actually the same strain. So even experimental differences in your assembly can result in sort of 5 locus differences at core genome MST. So the sort of nuts and bolts of what I guess most of you will be interested in was actually annotating isolate records within the databases. So we host annotated isolate and genome data. So if we look at a particular isolate record, and this will vary a little bit depending on which database you're looking at. All the databases will have slightly different fields set up that are appropriate for that particular organism, and they will have different schemes set up. But it all fits the basic pattern. So if you look at an isolate record, then any particular isolate may be part of a project. So, and it could belong to multiple projects. So we have information about that describes those projects. Then you have your section based on sort of your provenance or your primary metadata. So as I say, that's country, you've got year, you've got some information about disease, the biological source of it, and then you might have some serial group information. We also have accession numbers that link out to other data sources. So often like the short read data will be available and will have been uploaded to the ENA. What's that? Oh, okay. Would have been uploaded to the ENA, so that will be hyperlinked out, so you can get back to the original data. Tells you who has sent it, the last person to have touched that record, to have made any changes to it. So in this case, it was an automated tool. And then we record any changes to the record as well. So you can see who's done what to which record and when. And then you see these databases have been going quite a lot of time. So this was put in sort of 20 years ago and has been updated quite a lot in the meantime. Then you may or may not have this secondary metadata section. The reason we actually differentiate these is because of the way we store the data within the database. So the primary metadata tends to be fields that most records will have a value for. So that's things like country, year, etc., and all the basic housekeeping fields. And the way that's actually stored in the database is very much like an Excel table. It's just a column for each of those fields. But you can imagine if you have some data that's sparse, so you might only have information for maybe a few hundred out of tens of thousands of records in the database. It's actually quite wasteful to create a separate field within the table. And in fact, there are limits to how many columns you can put in a database table. And so we store these in a different way, in a sparse table, where basically we define it. This is an implementation that you don't really need to care about, but it's within the database, it's basically a 3-column table. We have the isolate ID, the field name, and the value. And that allows us to store any number of arbitrary extra values without only using space for data that you've got and not reserving space for all the records that don't have that information. So that comes under secondary metadata. And we can also set up some formatting for some of those fields. So for instance, sometimes it's possible to deduce information from a genome. And so for the Neisseria database, we do a test to see whether that genome would be covered by particular vaccines. The vaccines that have different protein components. We have a tool that can check to see the sequence of, or whether that sequence exists in the genome, the exact peptide sequence of that, or the translated peptide of the gene. And we also know what exact variant is present within the vaccines, and also how cross-reactive it is with other variants. And we have a whole list of which ones are cross-reactive and which ones aren't. And we're able to sort of make that assessment and then present this as a traffic light system for clinicians so that they can know that this particular isolate is or is not covered by a particular genome. So we can do all sorts of extra formatting, and we sort of hyperlinked to notes that describe exactly what's been done there. Then we can link out to publications. So you can link any number of publications to an isolate record. And you just do that by storing the PubMed ID with the record. All the other information is retrieved automatically from NCBI, from the PubMed database. And then so once that's pulled in, it goes into a separate sort of publication database, which you can then link to within the web interface, and you can query by author or year or whatever. And you can also, you get the full abstract, and all other isolates that have that PubMed ID will be now grouped within the interface as well. So you can return data sets from a particular published study. So this is quite a nice way of grouping sort of related isolates. Then we have information about the sequence bin. So these are your contigs that have come out from your Illumina assembly. So you've got your basic, fairly standard stats that you would get from that. So this is your number of contigs, total length, the max and mean lengths of those contigs, and then values about N50, shows you the distribution of the size contigs. And a link out to the sequence bin itself. And then from that, you get some nice charts of the breakdown of contigs. And also an integrated genome viewer. So you can actually see the loci that have been tagged within the database. Then we have the clusters, which we've already sort of briefly shown. And then the schemes that have been defined for that database. Now, a lot of databases will just have MLST and maybe core genome MLST. The ones that we tend to work on are getting more and more loci. So certainly Neisseria, which is the most developed, but also now Haemophilus and Strep that Odil's been working on have got a lot of loci which have been put into schemes. And we can group these schemes in a hierarchical way. So we can set up schemes that sort of reflect the biochemistry of the organism. So Odil's sort of capsule screens, we can sort of click that node there, and it expands out to sub schemes. And we're collecting information about those, about the loci that encode the enzymes within those schemes in exactly the same way as we do for MLST, just using allele numbers. So the ultimate aim is that we sort of can get some sort of prediction of phenotype based on the genotype. Some way off, but you've got to start with actually indexing the variation in the first place. And then we have some links to various tools that you can do on the sequence. So check the species ID using a ribosomal MLST tool, and you can also do an in silico PCR reaction based on that genome sequence. Test different primers to assess what sort of what products you expect to get from a PCR reaction. And then the database, the normal front end for the database is basically a query form. And the query form that you get presented with when you first go in, it's just like a single field and a drop-down list of fields and somewhere where you can enter a value. But this whole form is very customizable. You can expand individual elements. So if you wanted to add multiple provenance field searches, and you just sort of click this plus button, then you can have up to 20 different ones. But then you can modify this form. If you click this wrench icon, then it gives you a form that allows you to add other elements into there, so that you can do queries based on the alleles, particular alleles for certain loci, based on sequence type and clonal complex. You can even search on whether particular loci have been tagged. Or there are these filters which are built in. There's some which come predefined. So for instance, like the publications. So this gives you a searchable list of publications, and you can search that based on author directly from within this filter. Your project list, and you can select multiple projects. It's not just a single item. And then you can maybe search on clonal complex. And actually, this is slightly out of date now. The sequence bin, we now have a separate form element that you can query for that. And there's multiple things about the sequence you can now query, such as GC content and number of contigs and what have you. And you can add to these filters as you wish. So you can just, from the drop-down list, you can choose any field in the database and add it into the list of filters, and then that gives you a drop-down list of values for that filter, which allow you to do a quick query based on that value. And however you customize it will be remembered so the next time you go into the system. So you can build up complex queries based on any attribute within the database. And then once you do that, you click search and you'll get your results table. And there'll be certain fields get displayed by default. These tend to be the ones which are most useful, which I've deemed most useful. Other people might disagree, but if you disagree, you can customize it and set whichever fields you're interested in displaying within that data set. And all of these records are hyperlinked, so you can click the hyperlink and it will take you directly into the item record that we just saw. So once you have that table of results, then at the bottom of that table of results will be linked to all the different analyses and export functionality that exists within the database. So these exist as plugins to the software. So these are sort of individual bits of code which can be added into BIGSdb, as a sort of modular unit. And we have breakdowns. They really fit into these sort of three categories. So we have breakdowns where we can look at single fields. So you can sort of plot all your countries, for instance, on a map, see how they're distributed. Or look at any table, pie chart or bar chart or whatever. You can do a two-field breakdown. So this is sort of analogous to an Excel pivot table. So if you wanted to look at how capsule group breaks down against clonal complex, then you can get that out as a sort of as a table with clonal complex down one side, capsule group down the other. Your numbers, so that's very useful for looking at trends. You can look at you can choose any number of fields or loci and look at the number of unique combinations of all of those fields. So for instance, if you were designing a vaccine and you had some loci in mind, you could say, okay, I just want to look at these loci, tell me what combinations I see within that data set, and it will tell you all the frequencies. And so it's quite useful for doing assessment if you're designing vaccines, but also useful for actually lots of other things as well. And you can just pick one field. If you just want to export the ID numbers, for instance, you can just set ID as your combination, and it just gives you a list of IDs. And then there's tools that sort of break down like the sequence bin. So it gives you just a table of stats for the sequence bin. And that's exportable in Excel formats if you want. Then you can export the data in various ways. So your isolate data set, again, you can pick which fields you want. That can be all the loci as well, all the allele designations, and that will export that as an Excel file. If you want the sequence data, then you export all the contigs as FASTA files. Again, you can select multiple ones. You can select 1,000 or so at a time, and it exports them all in an archive file. Or you can choose certain loci or schemes that you're interested in and export everything as an aligned sequence. So, for instance, if you wanted to do a phylogenetic analysis where you're actually looking at the nucleotide sequence rather than the allelic profile, and you might say used MLST for that, then you could select all the MLST loci and it would align each locus in turn and then concatenate them all together. So essentially you end up with an alignment that reflects a particular scheme. So if you do that on a core genome MLST scheme, then you end up with a core genome alignment that contains all the coding sequences. So sometimes it's more appropriate to do a phylogenetic tree using that method than to look at the method I'm going to show in a minute, which looks directly at the allelic profiles. Just because of the assumptions that are made in the different methods. And then we have various analyses tools. So we have Genome Comparator, so I'm going to show that. That was really sort of the first genome analysis tool that we had within the system. Grape Tree, it's a really nice tree for looking at, for drawing a minimum spanning tree, and it works on tens of thousands of isolates, but it allows you to overlay metadata onto that tree. It gives you a nice sort of visual, and you can see how different features fit within the phylogeny of the organism. PubMLST is a little bit similar to Grape Tree. It's sort of a minimum spanning tool. Some of these methods upload to external sites, and some of them are integrated directly into the software. So Grape Tree has been developed by somebody else, but the code is available and we've incorporated it. Things like PhyloViz, ITOL, the interactive tree of life, and MicroReact are all external sites, but you can select your data set and then upload that data set to those sites and then visualize it within those sites. And I mentioned, so in silico PCR is another test that you can do. So just to give you an idea of how much this is being used by other people. So I've really been talking about BIGSdb. Now that is the platform, that's the software. We developed BIGSdb in order to support PubMLST, which is the website, and that's the collection of databases. There's actually a lot of confusion. People seem to think BIGSdb is just one database, and I think it's because it's got DB in the name, but it's not. It's a platform, and there are lots of different BIGSdb databases. So PubMLST itself is quite a large website now. We get about 20,000 unique visitors a month. Over 2,000 people submit data to it. And there's hundreds of curators all around the world who look after different databases on the site. It's not just us that use it. I mean, we write the software, but it's freely available for anyone else to use. It's open source software. So the Institut Pasteur use it to host their databases, but it's also hosted, other people use it. So for instance, like Pfizer and GSK run this internally to look after their data. And we have databases for, excuse me, we have a database for a lot of different organisms now. I think it just hit 130 different genera and species. So each organism has its own database. So they are completely separate databases. And for some of them, we have the whole genus. So for instance, Neisseria contains all the different Neisseria species within it. Others are just specific to a particular species. And then if we look back to when we actually started doing this, the design and initial work for BIGSdb started back in 2008. So it's had quite a long history of development. And actually 2008 was before we actually got genome sequences. So I've been working on databases for say MLST since I published MLST DBNet in 2004. So we're working on it for a couple of years before that. So really about 20 years. But it was 2008 that Martin Maiden, who's my boss, came to me and said, genomes are coming, we can't handle them, we need to be able to handle these when they become available. So the old software, you used to set up a database and you would decide in the beginning what loci you wanted to work on. It'd be like 7 or, yeah, normally 7. And that was it. That's all. You couldn't modify it easily afterwards. You couldn't just flexibly add new loci, and it certainly couldn't handle genome sequences. So it was really designed from using a lot of the same concepts that we had been using before, but with the idea that it would store genome sequences directly within the database, and you would be able to add any number of loci and organize those loci into any number of schemes flexibly sort of through the web interface, not relying on sort of a particular database structure that you decide at the beginning. Yes, and so it's really been my day job ever since. So it's kept getting employed so far. Long may that continue. And yes, as of this morning, I checked, it's now been cited 1,774 times. So quite pleased about that. I'd like it to get to 2,000. There'll be a tweet when it gets to 2,000. Yeah. And part of that is because we put that on the website and say, if you use this in your publication, then cite us, which is fair enough. But it's slowing down now because we have a new publication, so that one tends to get cited now. But a lot of citations tend to be viral. People copy what they've done in the past or what other people have done. So it's still being cited almost as much as a new paper, which is nice. Anybody interested in sort of how in the actual software, the actual languages and stuff? I'm not going to dwell on this, but the backend database is PostgreSQL, which is it's an open source database. It's very powerful. It's just as good as Oracle, but it's freely available. Most of the backend code is written in Perl, and the front end is HTML5 and JavaScript. And we use various different JavaScript libraries. So, for instance, like D3, you may have heard of, which is very useful for interactive visualizations. And it all runs on Linux, the actual server. But we're actually just thinking about that. The server is not just one server. It's split. The different functionality runs on different machines. So the database is one big machine, and that is a big machine. That's like 120 cores and a terabyte of RAM just to run the database. And then we have a web server, which is separate. And we have a different server that runs the API, the application programming interface, which can be used by third parties to access all the data. But you can have any number of, say, web servers that all interact with this one database. Right, so how do we use BIGSdb to compare genomes? Well, the first tool I'm going to talk about is Genome Comparator. So this was developed, this was really the first one that we sort of looked at. Basically, this came about from a conversation I had with Xavier Nassif when we were teaching on one of the Wellcome Trust courses at the Sanger Institute. I can't remember when, it must have been maybe 2009 or something. And he had a laboratory-acquired case of meningitis in his group. And he managed to get a blood culture of the bacteria that infected his co-worker. And of course, they knew the strain that they were working on, and he wanted to know, well, what differences were there? It's a natural experiment. To know what sort of differences we could find within the genomes from the strain that had been plated out within the laboratory and the one that had been recovered from the worker. And at that time, we didn't have any way of doing that. And so over a very sort of hectic weekend, with the very first version of Genome Comparator was written. And we were literally only looking at 2 genomes and comparing them against a reference. But the way it works is that you, from an annotated reference sequence, you take every coding sequence, every gene that's defined within that annotation, and then you blast that against each of the genomes that you're interested in. And if the locus, if the sequence you get back for a locus is the same as a reference, then we call that allele 1. If it's different, we call it allele 2, and so on. And so for each genome in turn, you look at it. And if it's the same allele between different genomes, then they get the same allele number. And so with this, you were able to build up an ad hoc whole genome MLST table without actually having had to define a scheme in advance. So you'll see here, the reference genome is all allele 1. And then so each column here now is a different genome. And so you see all these genomes have the same allele as the reference, except this one here, where it's a different allele. And in order to see like how much variation there is within loci, we change the color slightly as we find more variance. So you can actually just scan the table by eye and get a very quick feel for which loci are variable or not. And then some loci are incomplete. So in that case, we sort of mark those with a green eye. So that's just where it's fallen off the end of a contig. We can't retrieve the whole sequence. And then sometimes we can't find the sequence at all within the genome. So that's a missing loci. But we can treat that in exactly the same way as an allele. Okay, so if one genome has a sequence and one doesn't, then that's an allelic difference. So that's still useful information. The system can now work in two ways. It can either use the ad hoc method where you take an annotated reference, or you can just do it based on an existing scheme. So if you have a core genome scheme, it makes sense to use all the alleles that you've already scanned and stored within the database for the loci, rather than grabbing them from an annotated reference. So it will work both ways. And then once you've done that, once you've scanned it, and this used to take a long time because, as you can imagine, because the first version was very naive, it scanned each locus in turn, did a separate blast query for each locus. And that was fine when we were only looking at 2 genomes. It still took an hour, but it was fine. Trouble is, if you make something available on the web, people will always try to push it. They'll always, the data sets get bigger and bigger and bigger. And it gets to a point where either you can't support that, you have to limit what people do. And we used to put a limit of I don't know what the early limit. For a long time, we limited at 1,000 genomes. But doing it that way, some jobs would take 3 weeks to run. And so you can imagine if you had multiple jobs running that take 3 weeks to run on your server, you could never reboot your server because there was never a time when it wasn't busy. And if you did reboot it, you would cause somebody to lose 2 weeks of analysis. And of course, as databases get bigger, everything just slowed down as well. So again, we use as much sort of use. It's now all multi-threaded, and we scan all loci in one go. So it's much quicker. So now even 1,000 genomes you can do in a couple of hours. But what you get out from that list of alleles and the allele numbers is you can do a count of the number of loci that differ between every pair of isolates within your data set. And from that, you will get, it will generate a distance matrix whereby you can, so you can imagine this as actually as a square. You only showed a triangle so that you only shows the same numbers once, but the same isolates will be going along in the columns. And so, for instance, you can compare if you compare the second row to the reference, you'll see that there's 626 locus differences between that. And so then between these first two, there's 651 loci. Okay, so it's a very simple count, and computationally, of course, that's very simple to do. It is literally just counting how many differences there are within that matrix. So it's not having to look at sequence. Once you've done the scan, it's not having to do anything clever about looking at what those nucleotide differences are. And so this process is pretty quick. And you could just scan this table by eye and see which isolates are most similar. These two here, you'd have to work out exactly which column corresponded, but there's 34 locus differences here, 26. So these are very closely close together. And within Genome Comparator now, it exports everything in an Excel spreadsheet, and it does color those so you can scan the table a bit easier. But this particular matrix is in Nexus format, and we can use that. We can put that into an algorithm to display. But before I do that, I just want to sort of give you an idea of how you can treat allelic differences, and you can treat them very much. You can treat genetic distances in the same way as you treat geographic distances. Okay, so if you think about, I don't know if anyone here has ever used a road atlas, but in the days when we used to, before GPS, and you had to buy all your maps in a big book, on the back of that big book would be a list of cities and the distances between those cities. And it used to be in this sort of very similar to this actually. So I've done the same thing here. I've created a distance matrix based on distances between United States cities in miles as the crow flies, a direct straight line, not following roads and stuff. And we can take that distance matrix and we can put that into an algorithm like NeighborNet, and it will generate a map for you purely based on those distances. And there's no manipulation done into this at all, but you can see that map exactly matches the geographic map. All those cities are in their correct location. And the only change I had to do was just rotate it just to fit the map, but nothing else would need to be done. So it can determine that purely based on distances and trying to work out how they all fit together. But you could take your genetic distances, your allelic differences, and you can put that into exactly the same algorithm and then use that to find out which strains are most similar to each other. So this is a paper we did that looked at a university meningococcal outbreak using the same method, and you'll find, you don't need to go into detail about the particular strains, but there were actually two different strains that were causing disease at the same time. But we can, these are very tightly clustered together, but we were able to differentiate those from other isolates, which were actually of the same clonal complex, but we found within the carrier population, but they were actually very different. And we had a reference sequence in there that had been isolated a few years earlier. And so the differences this reference here was was isolated a few years earlier than both of these two. So we could see that these two were not at all related within that outbreak. They were just happened to be there at the same time. Okay, so that's Genome Comparator. Then the other tool I mentioned was Grape Tree. So this has been developed by Mark Arkman's group as part of the EntroBase database, which is sort of largely Salmonella specific database. And the idea of this is to handle huge amounts of data. They say 100,000 genomes at core PubMLST. That's what the paper says. If you ever try it on that, then your machine grinds to a halt and you can't really do very much with it. 50,000 is just about possible, I think. But it does work on a large amount of data. But it gives you this minimum spanning tree, which is very good for looking at closely related things. It's not, you can't make too many inferences about how things have evolved. I mean, if you look at this naively, you might think everything's evolved from this central clonal complex and it's all spread out. That's not what that shows, okay? It's very important to realize that. The whole idea of minimum spanning is that you try to join things using the smallest number of lengths of branches to do it. It's an algorithm that was originally developed for laying out telephone networks using the smallest amount of copper back in the Czech Republic, the very first algorithm. But it's been applied to lots of different things, but it is useful for showing you the most parsimonious links between things. But so things that are close together tend to be related, but things that are coming out from a central cluster are not. It's not showing evolution. Okay, but what you can do from this minimum spanning tree is overlay any of the metadata within the database. And so this is a tree that's been developed using core PubMLST. So this is your 1600 loci, Neisseria meningitidis. But then we've colored the nodes based on 7 locus MLST clonal complexes. And what it shows you is that 7 loci MLST is actually very, very good. Is able to actually recover most of that population structure. You know, it all makes sense. But you can use any of the fields that you're interested in. So you know, if you had years here, you can color these nodes. You can choose to show them as categories, in which case they'll use different colors that are as different from each other as possible, or you can show it as a gradient. So if you're looking at sort of age data, so years, then you're showing it as a gradient. You can see within the tree which clonal complexes are seen most frequently at different times. So there's all sorts of different ways that you can sort of play around with that data using Grape Tree to explore trends. And you may not have a preconceived idea of what you're looking for, but you can export, you know, all your metadata and then play around and see what falls out of it. And you can also drag and drop the different nodes within the tree, and it sort of all moves around on your screen. So it does produce some nice graphics that are actually sort of publication quality. So it's quite, and you can export them as SVG files. So it's a nice tool for producing nice images. So I've talked about genome clustering using core genome MLST. So I think this is almost the last thing. And so we have, as we know, we have core genome MLST schemes for different organisms. I'm not sure where we are with the strep pneumo one. It's coming though, isn't it, at some point? But so we first did it with Neisseria meningitidis. So that was Holly Bratcher sort of developed that. So this is a scheme for 1,600 odd loci. And there's various schemes for other organisms. And with these schemes, it's just basically you're taking your core genes and you're removing any loci that are what we call difficult, anything which are very similar to something else. It might be a paralogous locus where you might get more than one hit, or gene duplication, or anything that doesn't assemble well because of repetitive regions. So they're all core genes that behave nicely, essentially. So 1,600 loci. And we can, from that, we create core genome MLST scheme, and we define core genome sequence types just like we do standard MLST. So every unique combination of alleles across those 1,600 or so loci will get a different core genome ST. And of course, because only one locus difference is the new core genome ST, almost every genome will be different at that level. But you also have the complication when it comes to genome assemblies, in that you very rarely will get a complete profile. However good your assembly is, some loci will likely be missing. And that shouldn't be because they're missing actually in the genome, but it's probably some technical artifact that they haven't been recovered from the assembly. And so we have to have a way of being able to handle that. So we can define profiles which have an N in any position, and N just means any. It means we don't know what the allele is. And so in pairwise comparisons, we essentially are ignoring that position, okay? Because you're saying it might be the same. Well, actually, we are treating it as though it is the same as the one we're comparing against. So in this case, these two profiles here, this bottom profile has an N at this position, this top profile has an N at this position, but as far as when you're doing your comparisons, these two profiles are the same. But of course, there is a natural consequence of that, in that you can't have too much missing data. Because if you do, then everything becomes the same. You think of an absolute extreme case where you might have 1,600 missing data, it would be the same as everything else in the database, and everything collapses down into a single node. So you have to limit it, and we allow profiles that have up to 50 missing loci. That's a completely arbitrary number, but it seems to make sense and not cause us any problems. But it's just something to be aware of, that some profiles cannot, or some genomes cannot be assigned a core genome ST if we can't recover enough alleles. The other consequence of this is that some isolates might have more than one core genome ST, which seems a bit strange. But if you think about it, if you're defining each of these as a separate core genome ST, and essentially these are identical, then an isolate, if an isolate goes in, say, and it has, suppose it has a complete profile and it gets core genome ST1, second isolate that goes in, it might be missing 5 of those loci, but it's otherwise the same. You can't call that second isolate core genome ST1 because you don't know what those other 5 loci are. So you can make a choice. You either say, well, because it's missing those loci, but it's otherwise similar, I'm not going to give it a sequence type at all. Or you give it, you call it, or you do give it a sequence type, in which case you call that core genome sequence type 2. Trouble is now your first isolate that you gave core genome ST1 will also now exactly match that second one. So that's suddenly now got 2 core genome STs, which looks a bit messy, but it depends. It really matters as to what you're using those core genome STs for. And when I first did this, I thought, oh, I don't want all these different core genome STs. And we didn't allow you to have to define a second profile. But that meant, but then when we wanted to cluster, it meant you couldn't cluster some of these isolates. So the pros and cons basically are, if you want to cluster everything, or as much as possible, then you need to allow things to have more than one core genome ST, possibly. It doesn't normally happen. Most things only have one, but it's just something to be aware of, that you can see things. 2 core genome STs doesn't mean that it's contaminated. It doesn't necessarily mean that. It can mean that. But it can just mean that there's other profiles that are similar, but that have been defined that are missing some loci. Okay, does that all make sense? So when we want to cluster these to decide which isolates are similar to others at these different levels, we create what we call classification schemes, which are basically threshold groups for the core genome MLST. And so, as I mentioned, the thresholds we choose can either be arbitrary or they might have some rationale behind them. But for Neisseria, we've got 200, 150, 20, and 25. Well, sorry, for Meningitidis, we have those. For Gono, we've got slightly different thresholds. But the way the clustering works is that we, as you look through your data set, your first isolate goes into a group, and the second isolate you look at, if that is more than the threshold number of differences away, it goes into a new group. Okay, at each of those thresholds. Third one, and so on and so on. So every group that you get is at least that many differences apart. Okay, there's no isolates that are less than that between those 2 groups. What then happens is, if you get a new isolate that comes in, but it's within the threshold difference of 2 groups, then, okay, so where does that go? Well, it has to go somewhere. So what happens is it goes into, it tends to go into the biggest group, but then we merge the 2 groups. So everything that was in group 2 now goes into group 1. So this is why we call it single linkage. Those 2 groups have now been merged together by that single isolate that have come into the system. Okay, so that group 2, everything that's in group 2 go into group 1, and group 2 is sort of removed from the system. So what that results in is that over time, your groups get bigger and some groups merge. And that does mean that it's not stable. You can't use this as a stable nomenclature, even though people try. And generally, the larger groups don't change, but they can do if you make some sort of change in the database. But essentially, that allows us to know which core genome STs are grouped together. And because we know which isolates have which core genome STs, we can also determine which isolates group together. And so within the database, we can say within an isolate record, you can look at, say, if we look at this 25 locus threshold, we've seen there's 17 different isolates in the database that were within that threshold. And if we look at those isolates, you'll see that they all look very, very similar. They're all the same capsule group, they're all the same clonal complex. They've got the same antigenic profile. In fact, they're almost all from a similar region geographically. But none of that information has come from, it was used to make that assessment. That's all just come from the core genome cluster groups. Now, we do have various ideas as to how to make this nomenclature stable. There are things you can do. We don't do them yet. So one idea is Entrobase. Single linkage clustering is pretty standard method of clustering things, okay? It's sort of the go-to method. So the way Entrobase does it is they will run the clustering in training mode for 6 months or so, and then they say we're not going to merge groups any longer. So anything coming in it will just go into one group or another, but they then don't merge, and that gives you a stable nomenclature. But it does mean that over time your cluster it doesn't work as a clustering sort of thing, but it's still useful. We do have another method that's coming that that has been developed, but we are going to implement in BIGSdb that uses LIN codes. I don't know if anyone's come across life identification numbers. But it's basically, I can't really go into it, but it's basically a way that the life identification numbers uses whole genome ANI, average nucleotide identity. And it basically compares one isolate against another, but you can use this, and it's stable. You add more data into the system and sort of things don't change. We can map core genome STs onto, use the same idea, but use the core genome threshold groups in the same way. We just got a preprint out of it with Sylvain Brice. They've developed it. We got on the paper because we were going to implement it. So we will be looking at doing that. Okay, so yes, so you can use the same cluster groups form as I showed earlier. If you have a genome sequence, paste it in, you just want to not look at isolates in the database, but from your own genome, find out which sequence type, which core genome sequence type it's most similar to, and therefore which other profiles or isolates in the database are most similar to that. So that's all using those classification groups. Okay, almost done. All right, last thing, what's next? The only thing I'm going to talk about are the dashboards, analytic dashboards. So they are, well, they're sort of ready now. The whole idea about dashboard, well, the whole idea of why we wanted to introduce dashboards is that in order to use the databases currently, you need to have some sort of idea as to how they work. You know, how to use a query interface, and what information you might be interested in actually retrieving from them. And of course, if you're just a casual user, you have no idea about any of that. And so really the idea of this is that a dashboard provides sort of a way in for anybody to get some information from the database without having any preconceived information or any idea as to what's in there or what they might be interested in. So basically, these are an alternative front end to the database that break down all based on individual fields, and show you sort of what's in the database, but also gives you a way to so you can identify trends, for instance. So you may not know what capsule groups are common within the data set, but this you just scan your tree map and you get an idea straight away. And likewise, you see exactly which countries are submitting data to the database by looking at the map. But then you also have information about sort of your genome size, your distribution of genome size, and when things have been submitted to the database. And this whole the whole dashboard is completely customizable, so you can go in and you can add fields that you want to add to the database. But it also gives you a way of sort of educating users as to how they can construct queries, because for elements that break down sort of an individual field value, it has a link out to takes you directly to the data set for that value, and actually creates the data query. So a user will go in and say, click that link, and it will show you how you would query the database to search for Neisseria miniatis, for example. And you can also filter this data search based on different times. So you might only be interested in like the last year rather than all time. So you choose exactly which bit you're looking at. So this is sort of ready now, and it has been introduced. It's had a bit of a soft launch because you never quite know whether everything's going to work right. So it's available on Neisseria and Strep pneumo and Strep galactii and Haemophilus, but not as the default option. It's there as an option you can choose by sort of clicking the sort of top right link. But within a couple of weeks, we plan to make this live for all the databases and be the default option. And this is really the front end. So this is just a front end dashboard, but we do intend for this also to be used elsewhere within the site. So that, say, for instance, once you've done a data query, rather than just getting a table of results, you actually get a dashboard of results. So you can be able to interpret your query, in exactly the same way. And that would just be an option. You would still get to the table, as well. So that's all coming, and we plan to also have ways that you can drill down into the data directly from the dashboard. So all of these are sort of single field breakdowns because they have to be generated live from the database. So it has to be a quick data query. If you try to do complicated things like breaking down a capsule group against serial group or something, that's a much more complicated thing. So it can't return that information in real time. You can do it in near real time, a few seconds, but not the few milliseconds that we need for it to do for the dashboard. So that's coming, and that's all I have to say. So I'll stop.